Hello and welcome. My name is Therapy Snack, and today we're going to be covering off on a fairly sensitive topic. I'm going to be talking about suicide and suicide attempts, which I'm sure will do absolutely smashingly on the algorithms. With that said, obviously my area of focus here is not going to be anything akin to a motivational talk or a self-care or healthcare PSA. You can find a lot of that online. There's plenty of resources available. And unfortunately, as you're probably going to see, it usually comes in the form of a fairly shallow mental health campaign or WikiHow style guide. And that's not how I roll around here or how I slither. And as a result, we're going to go into a fair amount of detail. With that said, if for whatever reason you've been directed here by an algorithm and you are suffering from things like suicidal ideation, I absolutely encourage you to reach out to whatever your nation's mental health or suicide prevention hotline is and get in contact with them because um, that's not really going to be anything I'm going to be covering off on here. My background is in behavioral and social science, and I want to give you something here that's more akin to if you were a psychiatrist and talking to fellow psychiatrists or a forensic pathologist who wasn't sugarcoating, um, who was speaking kind of in the language of data and empirical rigor. What you're going to notice this time around is that compared to some of my other work, this will be a little bit more scripted. The goal being largely just to keep myself on track and to make sure that I'm not talking around in circles for 40 minutes and wasting everyone's time. At the end of the day, though, with this topic, the reality is public conversations around mental health are often driven by a very low level lay understanding. And that's created a lot of noise, and you're going to see that that hides a lot of sophistication and complexity around the actual science. Suicide in particular has a habit of being treated as one unified thing, and the data definitely show otherwise. So what I want to do is kind of open up the biology, the neurochemistry, and the forensic detail. And that's going to mean looking at a lot of things that are fairly uncomfortable, including things like parasuicidal behaviors, that function more as signaling or what is sometimes referred to as attention-seeking behavior, low affect versus high affect, concealed lethal suicides that almost never present until it's too late, and kind of getting into why there's a lot of confusion in the public conversation about foreseeability of suicide and kind of tells of suicidal risk. And we're going to kind of get into the fact that all of these things we're discussing are not really points along a continuum. They're actually distinct, and the biology shows that, and we've known that actually for some time. So the goal of this talk is to give you an insider's look at that literature. So we're going to be doing some literature review here a little bit, and I'm going to synthesize more or less and summarize for the audience, that being you. We're going to look at things like CSF metabolites, cerebral spinal fluid metabolites, neuroendocrine challenge studies, genetic associations, the kinds of things that you're never going to hear in a public health campaign, but that matter if you actually want to understand who dies, who doesn't, and why prevention often doesn't seem to work very effectively. My obligation here isn't really to manage an individual patient. I'm not operating as a clinician, nor am I one, nor do I claim to be one. My ethical obligation is to the truth as someone who's training to be a researcher. And I think that everyone has that, including, quite frankly, citizens, the general public. We want to be honest with what the literature actually says, even if it's messy and challenges some of the kind of simpler narratives around suicide. So, kind of to tick things off here, if you are looking for platitudes, this really isn't going to be the place. What I'm offering is a dissection of the biology of suicide, and I think you're going to find the literature very, very insightful. Our agenda for today is pretty straightforward. I'm going to go over some introductory related content, going to touch off on monomodal thinking, going to go over some forensic and clinical typologies, then we're going to delve into some neurobiological pathways and explanations, and then a conclusion. So in this introduction section, I kind of want to just cover off a little bit more on the more detailed aspects of our agenda. There are three key points I want to highlight. The first is going to be suicide, then cryptic phenomenon, and then attempters versus completers. You might be wondering why we're kind of going into this level of detail. Isn't suicide kind of obvious? But these kinds of definitional and kind of structured approaches are very important because whenever we examine research, the first thing we have to be clear about is how concepts are actually conceptualized and operationalized. What do we actually mean by suicide, for example? What do we mean by an attempt? Are there different types of attempts? Do we care? And those kinds of definitions and those kinds of levels of granularity really do shape everything else. Suicide gets talked about a lot as if it's one monolithic thing, but in the literature, what we see called suicide often folds together some very different activities, and that can create problems in the form of overaggregation. It's how you end up with this vague category where everything is suicidal, or stealing some common parlance, suicide coded. But the reality is that suicide itself is a very specific phenomenon, and there are distinct behaviors that fall inside and outside of that definition. You might be wondering, why is this important? And that's because if we want to identify true biological indicators, we have to know exactly what behavior those indicators are tied to. Otherwise, we're just correlating noise. So when I say suicide or completer or attempter, I'm being very literal. These are technical categories with real differences, and in the next couple of slides, we're going to start pulling that apart. We'll start with definitions, then move into the idea that suicide is a cryptic phenomenon, and we'll get into what that actually means. And eventually, obviously, into the core kind of thesis here, which is the split between attempters and completers. So what's obvious right away, even from the prevalence chart here, is that we're not really talking about a single clean category. You can see it. Suicidal ideation absolutely dwarfs suicide deaths in every country. And even attempts sit way above completions in almost every single country on this chart. And they are definitely still above in every single country. So already, the term suicidal behavior is covering a constellation of very different outcomes, with massive narrowing as we kind of get closer and closer to the act of suicide. In the literature, suicide itself is usually defined very narrowly, generally along the lines of a fatal self-injurious act with some evidence of intent to die. But even that definition is not as straightforward as it looks. If you hand that to a medical examiner, the real question becomes, what counts as evidence of intent? A note? A method that's obviously self-inflicted? That's fine. But what about a single vehicle crash on an empty highway at 2 in the morning? There's actually, as an aside here, a whole body of work that actually argues a non-trivial slice of traffic fatalities are in fact covert suicides. Now, we're not going to go down that rabbit hole, but it illustrates the point. Even a supposedly clean definition gets very messy at the edges, and it varies by jurisdiction as well. When we get to the surrounding constructs, for example, active suicidal ideation, thoughts with a plan, a method, some structure, passive ideation, thoughts about death or wanting to be dead but without a plan, those are broad, but they are distinct. Add to that deliberate self-harm, parasuicide, non-suicidal self-injury, each of which has its own acronym. There's lots of acronyms. But they also have their own literature that accompanies them, and they have their own clinical profiles. And on the attempt side, you'll see that there's medically serious attempts separated by non-medically serious attempts. This is not pedantry. This is not creating categories for the sake of creating categories. This actually matters. And you might be wondering why. Well, because when you collapse all of this under the heading of suicidal behavior, or worse yet, just say that it's all just a form of suicide or along, a graduation to suicide, you create quite the analytic mess. You think you're finding predictors of suicide, but in reality, you're just averaging across groups that don't actually share the same biology or the same risk architecture. And that's how you end up with null findings or spurious ones, which we're actually going to get into at the molecular genetics level here in the next couple of slides. And again, just look at the numbers. The gap from ideation to attempt is massive. Even if you wanted to call it a continuum, the attrition alone tells you that there are steps, filters, discontinuities. And as you're going to see, once you bring in the neurobiology, those gaps aren't just statistical noise or opportunity. They are telling you that these groups are qualitatively different. So let's pause here and ask the obvious question. If suicidal behavior is not monolithic, where do these kinds of subtypes come from? Are they just theoretical inventions? Do they actually have a basis in clinical and forensic observation? The first place to look is psychiatric autopsy. This is a method where clinicians reconstruct the psychological state of the person who has died by suicide, drawing on interviews with family, medical records, and contextual data. These studies consistently reveal that not all suicidal persons are alike. The clinical presentations, the psychiatric histories, the immediate recipients, they are different enough that you can't reasonably treat them as a single phenotype. Even contemporary clinical frameworks acknowledge this. The Collaborative Assessment and Management of Suicidality, CAMS, one of the more widely used models in medicine, explicitly starts from the concession that not all suicidal persons are the same. It distinguishes types of suicidal states and emphasizes individual drivers. That alone tells you the field has recognized heterogeneity, even if the biological literature has been slower to catch up. Now, compare that to the average lay triage approach. Take every suicidal statement at face value, assume maximum risk, and err on the side of caution. For the public, that's actually quite prudent, but in clinical practice, or in a research or scientific sense, especially at the forensic level, you can't really stop there. You have to parse, was this a medically serious attempt or a non-medically serious one? Was it impulsive or planned? Is the individual presenting with high affect, volatile reactive, or low affect, detached, flattened, concealed? Those distinctions change both the risk profile and the underlying etiology, the underlying cause. And none of this is actually new. The recognition of typological diversity goes all the way back to Durkheim. His famous taxonomy, egoistic, altruistic, anomic, fatalistic suicides didn't have access to the same neurobiology, but made the same fundamental point. People kill themselves for very different reasons, and the pathways are not necessarily interchangeable. Durkheim was, of course, a sociologist, not a psychiatrist, and definitely not a biologist, but the insight is the same. Fast forward to modern biological psychiatry, and you see the same picture here with sharper tools. Man's stress diathesis model, which we'll get into a little bit, for instance, differentiates between trait vulnerabilities like serotonergic hypofunction, aka underfunctioning of serotonergic systems, serotonin, and state triggers like acute stressors. Forensic typologies mirror this. Clinicians stratify impulsive parasuicidal gestures from concealed violent completions. And when researchers actually respect those distinctions, the biological signals align fairly beautifully. Genetic polymorphisms, neuroendocrine markers, and circuit level findings map directly onto the clinical subtypes that psychiatrists have recognized for decades. That's the payoff. Typologies really aren't arbitrary. In fact, the goal is to ensure that to a certain degree they're mutually exclusive. They're historically grounded thanks to things like psychiatric autopsy, through qualitative case study analysis, and therefore they're clinically functional. And most importantly, they align with the biology when you stratify. The more precise we are with categories, the more the data stops looking noisy and starts looking very explanatory. So, cryptic phenomenon. When I say suicide is a cryptic phenomenon, I mean it in the biological sense. In biology, cryptic species look identical on the surface but turn out to be genetically distinct. Suicide works in many respects the same way. It looks like one category, but once you stratify, the biology says otherwise. Look at heritability, for example. Ideation carries a heritability coefficient of about 0.36, while attempts are closer to 0.17 once you control for psychiatric conditions. That's inverted from what you'd expect if they were just spots along a kind of severity continuum. This tells us these aren't just graduations of the same trait, they're partially distinct constructs. This becomes the same with 5-HTT-LPR. Broad studies come back inconclusive when we kind of aggregate everyone who's suicidal or has committed suicide into kind of the same cohort. But when you split violent from non-violent suicides, the short allele signal shows up in the violent subtype. If you ignore the subtyping, you never see it. And that's kind of the bigger point that I'm trying to get at here with cryptic phenomenon. At every supposed step of the continuum model, ideation, attempt, completion, we find discontinuities. Different heritability coefficients between ideation and attempts, different genetic signals between violent and nonviolent completers. The model of a kind of continuum theory, if you will, would say that these should kind of blur together. They should generally be the same, but the data says that they don't. So suicide as a single unified construct is like a cryptic species, morphologically the same, biologically distinct. Unless you stratify, you collapse meaningful differences into noise. Before we get into attempters versus completers directly, we need to stop and look at what I'll call the monomodal fallacy. The monomodal fallacy is the assumption that if two people present with the same observable behavior, then that behavior must have the same underlying cause. It's a form of category error, and in logical terms, you mistake a shared surface outcome for a shared etiology. Science is littered with this mistake, but psychology and psychiatry have been particularly vulnerable because so much of the field historically was built by categorizing constellations of symptoms without any underlying mechanistic evidence. You take a set of heterogeneous behaviors, you slap a label on them, and you assume that the label implies a singular mechanism. The classic case is hysteria. For most of the 19th and early 20th centuries, hysteria was the diagnostic wastebasket of psychiatry. It collected everything from conversion disorders to dissociative states, from what we now recognize as epilepsy to trauma-related neuroses, and even behaviors that weren't pathological but simply didn't conform to social norms. Freud famously theorized that hysteria was the product of repressed trauma or unconscious conflict. The implicit assumption was monomodal, one mechanism, one disorder. The reality, which only emerged after decades of careful work, was that hysteria actually wasn't a disorder at all. It was a miscategorization. It was an artifact of lumping together dozens of distinct pathologies, and in some cases, no pathology at all. The result was not only scientific confusion, but active harm. People were treated for a phantom diagnosis while their actual conditions, epileptic seizures, mood disorders, PTSD, even perfectly normal reactions, went unrecognized. That is exactly, to a degree, the danger with suicide. By collapsing ideation, gestures, non-suicidal self-injury, low lethality attempts, and high lethality completions into a single category of suicidal behavior, or a single concept of suicide, the field commits the same monomodal fallacy. The assumption becomes, these are all graduations of the same thing, a continuum model. But the data, as we've just seen, is already contradictory to that. Different heritabilities for ideation and attempts, distinct genetic associations between violent and nonviolent completions, neurochemical and circuit level differences between parasuicidal acts and lethal suicides. Ultimately, this arrives at the conclusion that these aren't graduations of a single mode, they're actually distinct biological and behavioral entities that look the same on the surface, just as hysteria once looked like a single syndrome until all the pieces were separated. The bottom line, if we let monomodal thinking dominate suicide research or our understanding and conceptualization of suicide, we guarantee we'll miss the signal. We'll chase an imagined universal mechanism that doesn't exist, and we'll keep producing null findings. The history of hysteria is the warning. If you don't break apart the wastebasket category, you end up studying a phantom. Alright, so let's get into the specifics of the genetics here. We've talked a little bit about this. I've brought up 5-HTT-LPR a couple times here, and I think it's good to kind of get into the nitty-gritty. Because this is really where the separation between attempters and non-attempters starts to crystallize, and violent versus non-violent completers also starts to show up. What you're looking at on the side of this slide is chromosome 17. More specifically, the locus is written as 17q11.2. For the sake of some education here, let's kind of break that code down because it actually does mean something. The first two digits tell us that this is on chromosome 17. The letter can either be P or Q that comes after it. P refers to the short arm on the chromosome, and Q refers to the long arm. In this case, it's Q, so we're on the long arm. What we've then got, obviously, is 11.2, and that specifies region 1, band 1, subband 2. In other words, this isn't just somewhere random on the chromosome, it's actually precisely localized to that stretch on the long arm. These designations are standard cytogenetic coordinates, and they allow us to specifically situate genes along a karyotype. The gene at this locus, so at 17q11.2, that we care about is SLC6A4, and that's not just the alphabet for you. That actually stands for solute carrier family 6, member 4. That name, again, probably looks like alphabet soup, but it actually literally tells you what the protein does. It's a solute carrier. In this case, it transports serotonin back into the presynaptic neuron after release. Functionally, we call that a serotonin transporter, in this case 5-HTT. This transporter is critical for clearing serotonin from the synaptic cleft, thereby regulating how long and how strongly serotonin can act on postsynaptic receptors. The specific polymorphism we care about here is in the promoter region, known as 5-HTT-LPR, hence the name. The serotonin transporter link polymorphic region. This is a VNTR, which is a variable number tandem repeat. Probably should explain to you a little bit what that actually means. So polymorphism simply means there's a variation in the sequence in the population. Multiple alleles are possible. A VNTR means that that variation comes in the form of repeated units. A short DNA sequence is basically repeated back to back to back, with the number of repeats differing between the alleles, or the genetic options, if you will, that we have for the gene. In this case, there are two common variants, the short allele, which contains about 14 repeated units, and the long allele, which contains about 16. That 2 repeat difference probably sounds fairly trivial, but it actually has major functional consequences. In research, we know that it alters the efficiency of transcription from the promoter. Promoter is involved in that extensively. Again, not going to get into the weeds on exactly how that works, but functionally, we've increased the promoter region, and would you look at that, the efficiency of transcription from the promoter increases. The studies actually give us a number for that. They consistently show that the L allele drives transcription 2 to 3 times more effectively than the S allele. More transcription means more transporter protein gets expressed because we're transcribing more, and therefore we pump out more of this serotonin transporter, and therefore we clear out serotonin from the synapse more quickly. So functionally, if you carry the L allele, your serotonin system leans towards faster reuptake and therefore shorter signaling duration. That serotonin doesn't stay as long in those synaptic clefts, those connections between neurons. If you carry the S allele, however, you have fewer transporters, slower clearance, and therefore altered serotonergic tone in circuits like the ventromedial prefrontal cortex and the amygdala. We'll get into kind of what that means, especially with the ventromedial prefrontal cortex, or what's sometimes called the VMPFC. We'll get into exactly why that's important, but what you need to know is that that's not a cosmetic difference. That's the kind of system level shift that changes how inhibitory control and affect regulation operate. So how does this map onto suicide phenotypes? Broad population studies of 5-HTT-LPR often come back as inconclusive. So if we aggregate everyone who's kind of all in a suicidal population or have committed suicide and we just kind of treat them all the same, we don't really find a lot. But what research has found is, very interestingly, when you stratify, when you start separating and categorizing out attempters versus non-attempters and violent versus non-violent completers, the pattern starts to become fairly clear. Attempters, and especially violent attempters, are disproportionately carriers of the S allele. Non-attempters don't show the same enrichment. That's the point. If you treat suicide as a monolithic construct, variation disappears in statistical noise. This 5-HTT-LPR mutation doesn't really seem to do a heck of a lot. But if you split the phenotypes, the biology really starts to line up. That serotonergic dysregulation starts actually making some sense and starts having an impact. So far, we've focused on serotonin, and in particular the transporter gene at 17q11.2, that HTT-LPR mutation. And that's foundational. It's probably one of the most well-supported ties to suicide. But the literature points to other processes as well, and we're going to want to explore those a little bit, specifically in the monoamine catabolism and neurotrophic signaling areas. So let's take a closer look at kind of two of the most common that are studied. There's many more, which we're not going to get into, but COMT and BDNF are kind of the two competing ones. So first, COMT, or catechol O-methyltransferase. That's the gene for COMT that sits on chromosome 22, and we're going to go give another coordinate set here, 22q11.1 to q11.2. Again, Q is for the long arm. We're on region 1, band 1, subbands 1 to 2. COMT does something a little bit different. Rather than going into the kind of regulatory regions like the promoter region, COMT encodes an enzyme generally that inactivates catecholamines, specifically dopamine, epinephrine, and norepinephrine, by adding a methyl group. So basically, this kind of functions as a form of biochemical regulation of neurotransmitters. This is one of the major ways that catecholamine signaling is regulated in the synapse. The key variation here, though, and this is what I was trying to get at a couple sentences ago, is that what happens is we have a single nucleotide polymorphism, known as an SNP, given the designation RS4680, which is just a code that makes it very easy to kind of track all of these mutations because there's a lot of mutations in the human genome. An SNP means exactly what it sounds like, so we have one base in the DNA sequence that's swapped for another. In this case, we have a guanine to an adenine transition, and that changes the codon, which is kind of a frame, if you will, of three nucleotides that changes. And this results in an amino acid substitution. So we actually turn valine to methionine at position 158 of the protein. This is why you'll actually see it called Val158-MET polymorphism. And again, you can kind of see that actually there's a lot of information in these somewhat seemingly very confusing strings of words and numbers. So Val158-MET tells us that we're switching a valine for a methionine at position 158 on the protein. Might be wondering, okay, why does this matter? We're swapping amino acids. That's very cool. Don't care. The reason is because this isn't just a regulatory variant like a promoter repeat, where we're like increasing or decreasing efficiency or blocking coding or those kinds of things. It's actually in the coding sequence itself, which means this is going into the protein. And again, we've talked about what COMT does. It is responsible for inactivating various different types of neurotransmitters at the synapse. It's part of neurotransmitter regulation. So this mutation occurs in exon 4, which means it's part of a coding sequence, and that means it actually alters the structure of the enzyme because we've changed one of the amino acids that make up the protein. Valine at that site produces a COMT protein with slightly higher thermal stability. Methionine makes it less stable. The functional consequence is that the Val-Val genotype produces about 38% more enzymatic activity than the MET-MET genotype. So the original, the valine, more enzymatic activity, meaning it's inactivating more effectively. The MET-MET genotype being less effective because it breaks down easier, lower thermal stability, causing things like denaturing or inactivation, or simply just not operating at peak efficiency. And this actually shows up in, again, enzymatic activity in the human prefrontal cortex. Heterozygotes, so Val-MET, fall in between, but those with, again, the MET-MET mutation are going to see far lower enzymatic activity, so far lower regulation in this kind of category. Functionally, higher COMT activity means faster degradation of dopamine in the prefrontal cortex. That translates into lower tonic dopamine levels, which has downstream effects on executive control, stress reactivity, and affect regulation. Lower activity, so the MET-MET mutation, does the opposite. More dopamine hangs around, but at the cost of increased noise and signaling and higher susceptibility to stress-regulated dysregulation. Both ends of this variation have actually been linked to psychiatric phenotypes. For example, schizophrenia, affective disorders, and importantly to us, of course, suicidal behavior. So that's catecholamine catabolism. Now let's look at neurotrophic signaling, specifically BDNF, or brain-derived neurotrophic factor. BDNF is a little different from our other two, which is that it's actually a growth factor. It's a protein that promotes the survival, growth, and synaptic plasticity of neurons. Also involved in things like depression. It's heavily expressed in the hippocampus and the prefrontal cortex. You'll notice that the prefrontal cortex is quite commonly referred to in a lot of this research, and we'll get into why in a little bit. Both regions, though, the hippocampus and prefrontal cortex, are implicated in mood regulation and suicide. The major polymorphism here is RS6265, also known as the Val66-MET polymorphism. Keeping with the pattern here, you will be no doubt surprised to learn that what we are doing is we're switching a valine with a methionine amino acid at position 66 on the protein. This kind of results in some changes in the substitution, which results in altering intercellular trafficking and activity-dependent release of BDNF. In other words, neurons carrying the MET variant release less BDNF in response to activity. So we've got a regulatory response control failure or dysregulation. So why does this matter? So this is because BDNF isn't just a background growth factor. It modulates synaptic plasticity, learning, and stress response, which you can imagine is probably quite important when we're talking about suicide. Reduced activity-dependent BDNF release has been associated with impaired resilience to stress, with higher risk of mood disorders. In the suicide literature, the Val66 MET polymorphism has been linked to increased risk of attempts, particularly in interaction with environmental stressors. The important point here is not to memorize every polymorphism. I assure you this will not be on the exam at the end of the video essay, but to recognize the pattern. We see signals in serotonergic regulation, SLC 6A4, 5-HTT-LPR, in catecholamine metabolism, COMT Val 158 MET, and neurotrophic support, BDNF Val 66 MET, different pathways and different mechanisms, but all converging on the same outcome. They alter the circuits that regulate affect, stress, and impulse control. Suicide then really can't just be put forward as kind of a serotonin story. It's polygenic, it's multimodal, and the more you stratify the phenotypes, the clearer these biological associations actually become. So up until now, we've been looking at individual genes and polymorphisms, 5-HTT-LPR, COMT, BDNF, and their functional consequences. This slide really puts all of that detail into a bigger picture, the neurobiological pathways implicated in suicidal behavior. Let's start with serotonin, because of course that's kind of the star of the show. One of the earliest and most robust findings in this field was low cerebral spinal fluid, 5-HIAA, hydroxyindoleocetic acid, which is the major metabolite of serotonin. A metabolite, for those that might be curious, is simply a breakdown product. In this case, what serotonin gets converted into after being catabolized. Decades of post-mortem CSF studies showed that lower 5-HIAA levels are consistently associated with suicide. This kind of gets us to the low serotonin thesis. And it's specifically found in completers using planned, concealed, and highly lethal methods. Now, this gets a little bit more interesting, though, because it's not, the evidence upon further analysis doesn't just point to low serotonin. Phenylphluramine challenge tests, which use a serotonin releasing agent to provoke serotonin release, show blunted prolactin responses in depressed patients with a history of suicide attempts. This tells us something very interesting, which is that insensitivity isn't just at the level of neurotransmitter availability. So it's not just an issue with producing serotonin, it's also receptor level rigidity. Even when we flood the system or have the system flood itself with serotonin, the downstream signaling still fails. This is why I emphasize that it's not simply just low serotonin. It's a system level failure of serotonergic regulation. Functionally, the critical consequence of the serotonergic dysfunction is disinhibition. The ventromedial prefrontal cortex, what I'm going to refer to as the VMPFC, normally acts as a gatekeeper, integrating affect, simulating outcomes, and imposing restraint. With low serotonergic tone and receptor level insensitivity, that gatekeeper function breaks down. The result is not cartoonish impulsivity. It's actually a subtle but catastrophic inability to interrupt escalating ideation, to forecast risk, to insert a break into the sequence. This connects directly to the epidemiological patterns we saw earlier. Think about the massive drop-off between ideation, attempts, and completion in every data set in that kind of cross-national prevalence rate assessment. Most people stop at the ideation stage, and one of the strongest reasons you're going to generally find when those kinds of people are interviewed is fear. Fear of botching an attempt, fear of pain, fear of permanent disability, etc., etc. In other words, inhibition. For the vast majority of people, inhibition is the wall that ideation never crosses. In those with serotonergic disinhibition, that wall crumbles. They don't spiral with anguish or torment, they move with flat affect, with concealment, and with lethal planning. The system that would normally interrupt the behavior simply doesn't function. Now it's worth pausing here, because this is what most people, even those with training in the behavioral sciences, this is where they kind of hit a wall. Everyone touched by suicide at some point asks the same question. Why? Why would someone do this? Why would they cross that line? And the difficulty in answering is usually because we underestimate the role of disinhibition. We try to get into the mindset to imagine the thoughts or emotions that would push a person towards suicide. But that's actually not the biological mechanism. That's actually not what's going on. The critical shift is not necessarily in the presence of overwhelming pain. Plenty of people feel that and never act. It's in the collapse of the gatekeeping function, the failure of inhibition. Once you see it that way, the paradox becomes clearer. The real question then isn't really why do people kill themselves, but rather why don't more people kill themselves, which is a fairly ominous question to ask. But the answer for most of us is that wall of inhibition holds. For a smaller subset, that wall is compromised, and the act that feels unimaginable from the outside becomes frighteningly available from the inside. Layer onto that the other converging biological systems, HPA axis overactivity, hypothalamic pituitary adrenal axis, cortisol dysregulation, often linked to early trauma, prefrontal cortical dysfunction or injury, and the genetic vulnerabilities we've discussed. None of these are necessarily sufficient alone, but together they map into a multifactorial etiology of suicide. That's what Mann's model in the image there is showing. It's not really one pathway, it's not one gene, it's not one transmitter. It's the interaction of a genetic diathesis, quite frankly. Neurochemical regulation, endocrine stress response, cortical circuitry, all of those things. But the through line is biological. The findings are therefore not arbitrary, they're not activist, and they're certainly not anecdotal. They're robust signals that explain why some people with ideation never progress, while others cross the threshold into high lethality completion. So now we come to the central division, attempters versus completers. This is really the cornerstone of the entire presentation. We've talked about genetics, neurobiology, typologies, funnels, everything. And we've gotten to this distinction. So let's begin with completers. In the biological data, completers are consistently shown to have low levels of CSF5-HIEA, the serotonin metabolite we talked about earlier. They also show blunted neuroendocrine responses on pharmacological challenge tests, notably phenethylamine challenges. For example, where normal individuals mount a prolactin response and completers do not. Imaging and lesion studies point to reduced activity in the ventromedial prefrontal cortex, as we've discussed, is the brain's kind of gatekeeper, integrating affect, forecasting outcomes, and imposing inhibition. Put all of this together, and you have reduced serotonergic inhibition, a compromised gate, and diminished risk forecasting. Behaviorally, completers therefore tend to kind of go towards concealment, planning, and blunted affect. Concealment is one of the strongest tells. If someone hides their intent, it is a very good indication that they are at extremely high risk. Planning latency is another marker. These individuals don't decide in a fit of rage and act within minutes. They decide, plan, and then later act, often with methods of very high lethality. Clinically, their presentation is low affect. They're not the people sobbing in emergency rooms or calling hotlines. In fact, many of them are oddly calm, sometimes even relieved once they've resolved on a plan. And that's what makes sense biologically. The gatekeeper's off, inhibition is down, and the act moves forward without the usual restraint. Now, attempters, and here's where it's kind of important to differentiate medically serious from non-medically serious attempts. I want to focus first on non-medically serious attempters in that kind of cohort, because that's where kind of most of the parasuicidal behavior sits. These individuals look very different in the data. Their CSF5-HIAA is higher, serotonergic function is intact, their neuroendocrine response to challenge are normal and often even heightened. Imagine someone showing kind of hyperactive ventromedial prefrontal cortex activity. In other words, their biology is not really the picture of inhibition collapse, but of reactivity and volatility. Behaviorally, they are high affect, they escalate quickly, and what we find generally is that their decision to action latency is typically within the span of 5 to 20 minutes. And that's really critical. They act impulsively, and they often regret it soon after. These typically show up in the cases where someone impulsively swallows a bottle of pills, for example, and then 20 minutes later panics and calls for help. But unfortunately, in some some cases that bioavailability is already kicking in. These are often the tragic regret suicides, where individuals did not actually want to die, but the impulsive act still proved lethal. This is also where parasuicidal behavior really kind of has to be addressed. Parasuicide covers deliberate self-harm, suicidal gestures, and non-lethal attempts whose functions are not really identical to lethal intent. The functions include affect regulation, relieving acute distress, interpersonal signaling, communicating need, influencing others' behavior, self-punishment, or even escape. It's important to note that this classification of parasuicidality is not a moral judgment. It's clinical descriptors of function, and psychiatrists, when speaking plainly, will kind of attribute much of this somewhat bluntly as attention-seeking behavior. And this isn't to trivialize it, but more to name its function. These individuals are absolutely in pain and they absolutely need treatment, and their acts are obviously dangerous, but the mechanism is distinct from the concealed, blunted, lethal profile on completers. And that distinction is very crucial. It changes how we assess risk, how we treat, and how we study biology. Again, there is overlap. Some completers have a history of parasuicidal acts, some parasuicidal individuals escalate and die, but in aggregate, the distributions are clear and robust. Completers cluster in the low affect, concealed, biologically disinhibited phenotype, and attempters, especially non-medically serious attempters, cluster in the high affect, reactive, parasuicidal phenotype. If you collapse them into one kind of wastebasket, if you will, category called suicidal, you erase the very real differences that explain outcomes. So now we're kind of at the final point here, which is implications. The themes here are totalization, monomodal prevention, and failure of fit. First, totalization. By totalization, I mean the intellectual temptation to claim we've captured everything, and that we have an all-encompassing, parsimonious model that explains suicide as a singular phenomenon. That's very attractive, because the act looks singular, one outcome, one fatal endpoint. But the biology tells us that's false. Everything we've reviewed points in the opposite direction. 5 HTT-LPR variations, CSF, 5 HIAA deficits, VMPFC dysfunction, BDNF variants. These are not one pathway, kind of converging onto one thing. There are actually multiple entry points into different pathways, some of which arrive at the same endpoint. Mann's diagram from the previous slides really drives this home. Suicide is not unifactorial, it's multivariate and multifactorial. And that brings us to the univariate fallacy. This is kind of the cousin of monomodal thinking. It's the belief that because a phenomenon looks uniform, that it must be driven by one cause. That error has plagued medicine forever. Hysteria was a perfect example. A wastebasket diagnosis that lumped unrelated conditions into one label. Suicide in much of the public discourse is treated the same way. One label, presumed one cause. But what we've seen here is that it's a cryptic phenomenon, superficially similar but mechanistically distinct. The implications for prevention, therefore, are actually quite serious. Current prevention infrastructure is overwhelmingly designed for the impulsive subtype. That's why so many strategies focus on means restriction, taking away the bridge, locking up the firearms, slowing access to medication. And there is a logic in that. When you erect barriers, you can interrupt impulsive, high-affect actors. And there's decent data showing that those deaths actually do go down, but the overall suicide rate doesn't collapse, and that might lead one to ask why. And that's because individuals in the low affect, concealed, disinhibited phenotype are not stopped by superficial friction. They plan, they conceal, and they switch methods. Economists call this method substitution, and the literature absolutely bears it out. Blocking one path and motivated individuals shift simply to another. That doesn't mean that means restriction is pointless. It buys time, it deters the most impulsive cases, but the ROI diminishes sharply as you move towards concealed completers. This is why you see flat suicide trends even as governments pour millions into surface level interventions. The target population isn't the one driving the fatalities. And that leads us to the second failure, collapse of monomodal prevention. Because we've treated suicide as one phenomenon, we've built one prevention architecture, and it captures the wrong cohort. Public campaigns, reach out, it's okay to talk, they overwhelmingly attract parasuicidal individuals, which is fine, those people need help, but they aren't the ones driving the lethality curve. Low affect concealed completers don't signal, they don't call hotlines, and they don't really show up in the ER screaming. They're absent from intervention almost by design. That's why actuarial psychiatry, risk assessment, and forensic and clinical practice is so aggressive. Clinicians know the real risk is often invisible until it's too late. They're trained to pick up on subtle actuarial risk markers, history, comorbidities, neurobiology, if available, precisely because overt cues aren't reliable. That actuarial aggressiveness looks harsh from the outside, but it's the only rational stance when you're trying to catch concealed cases. Finally, to tie this together, suicide is not a singular pathology. It's an outcome, a common final pathway produced by multiple distinct etiologies. When we stratify violent versus nonviolent, medically serious versus non medically serious attempts, ideators versus completers, the differences aren't cosmetic. They map to neurobiology, heritability coefficients, to receptor level physiology. They're real. So the implications are blunt. If we persist in treating suicide as a monolithic construct, we will keep building interventions that net the wrong population. If we stratify and respect the biology, we can target prevention where it matters. That's the collapse of totalization, the failure of monomodal thinking, and the next step suicidology has to take.